Mon nom est Jean-Éric Tremblay, je suis le directeur de l'Institut nordique du Québec et aujourd'hui, il me fait plaisir d'accueillir le professeur Mark Patterson. Marc va vous être présenté plus en détail dans un instant, mais je voulais vous donner un petit peu de contexte sur sa venue à l'Université Laval et le programme là, qui permet de l'accueillir. Donc, c'est un programme de la Fondation Fulbright. Qui est un organisme qui gère plusieurs programmes d'échange réciproque entre le Canada et les États-Unis. Au Canada, c'est parrainé par le ministère des Affaires étrangères et la Fondation Fulbright gère plusieurs bourses qui sont prestigieuses et très compétitives et qui sont offertes à différents niveaux académiques. Donc, il y en a pour les étudiants, les postdoctorants, les professionnels et les professeurs. Donc, je vous invite tous à suivre ce programme-là. Il y a beaucoup d'opportunités très intéressantes qui sont offertes. Dans le cadre de ces activités, le volet canadien de Fulbright a mis en place un programme de chaire en études environnementales et l'Université Laval, plus précisément Sentinel North, qui apporte un soutien financier, et l'Institut nordique du Québec ont proposé une chaire à Fulbright et cette chaire-là a pour thématique l'avancement de la recherche transdisciplinaire sur le Nord en changement. Donc, le webinaire d'aujourd'hui est organisé conjointement par l'INQ, Sentinel Nord, Québec Océan et ArcticNet. Et il a pour but d'accueillir parmi nous le professeur Patterson et de vous le présenter. Donc, Mark Patterson provient de l'Université Northwestern en Illinois et c'est le récipiendaire de la chaire pour 2022-2023. Deux professeurs à l'Université Laval sont particulièrement impliqués dans l'accueil et la collaboration avec Mark. Il s'agit des professeurs Ladd Johnson et Philippe Archambault. Donc, je vais laisser le privilège à Ladd Johnson de vous présenter Mark, puisqu'il le connaît depuis quelques années. Philippe a un petit empêchement ce matin. Normalement, il devrait se joindre à nous pour animer la période de questions. On ne sait pas si il va y être. Sinon, Ladd continuera pour animer la période de questions. Donc, je vous rappelle que pendant la conférence, vous pouvez poser vos questions sur le chat ou dans le Q&A et on va prendre les questions après la présentation du professeur Patterson. Donc, je cède la parole à Ladd Johnson.
Bonjour, je m'appelle Ladd Johnson, professeur en biologie à l'Université Laval. C'est un véritable plaisir de vous présenter Mark, professeur Patterson, aujourd'hui parce que je le connais plus que quelques années. On a fait d'énormément, c'est 40 ans maintenant. Parce qu'on se croisait pour la première fois à Harvard quand je postulais pour pas un poste, mais pour être étudiant là-bas. Mark est déjà en train de faire son bac, son maîtrise, ainsi son doctorat là-bas. Par la suite, on se croisait à quelques congrès parfois, mais j'ai suivi son parcours assez attentivement. Il a eu le poste en Californie, à Davis, l'Université de Californie à Davis. Par la suite, en Virginie, à la Virginia Institute for Marine Sciences. Et finalement, comme Jean-Éric a mentionné, il est poste actuellement en tant que professeur à Northeastern University à Boston. Donc, il a fait un genre de boucle de revenir à Boston pour on ne dit pas terminer son carrière parce que son carrière est juste à commencer à développer. Mais je suis particulièrement intéressant parce que lui, il est unique parce qu'il a fait un genre de mariage entre la science, la biologie en particulier, ainsi le génie. Donc, il a été doué dans les deux. On a ici une faculté de sciences et génie, donc ça va être parfait pour Mark, mais il a une poste dans la faculté de sciences ainsi la faculté de génie à Northeastern, donc parce que c'est si rare d'avoir quelqu'un qui était à l'aise dans le deux. Donc, j'avais demandé lui encore un peu d'où provient sa formation. Il est autodidacte, il a pris tout ça, a commencé en école primaire. Donc, encore, il y avait différentes choses. Et aujourd'hui, ils vont parler. Effectivement, quand il nous a approché pour la première fois, on a vu tout de suite encore une situation idéale ici pour Mark de s'installer ici, de prendre encore la biologie du Nord, ainsi encore appliquer la technologie que lui-même a développée avec sa propre entreprise qu'elle a démarrée dans les années 90. Donc, encore, je peux déjà parler trop, mais encore, j'ai hâte de voir que ce mariage de biologie et de technologie va nous donner peut-être dans les prochains mois cet été. Mark, la parole est à vous.
Oh. Merci pour l'introduction, Lad. Je suis très heureux d'être ici à l'Université Laval pour mon congé sabbatique. Un grand merci au professeur et président Stéphane Boudreau pour lettre d'invitation et aussi Martin Fortier et Phil Archambault et vous, mon ami de longue date, professeur Led Johnson, pour leur hospitalité et leur travail considérable qui a rendu cette aventure possible. Voici où je travaille. Mon laboratoire se trouve ici à Nahant, au nord de Boston. Là dit que j'ai une nomination conjointe entre le Collège de sciences et le Collège de génie. Aussi, je travaille avec l'Institut mondial de la résilience, situé sur le campus principal de Boston, où je travaille sur la résilience du réseau de smartphones et autres choses, des systèmes de communication d'urgence. Je suis un radio amateur depuis 52 ans et j'ai utilisé mes compétences dans de nombreuses situations extrêmes en Afrique et dans les Caraïbes et aussi pendant les ouragans aux États-Unis. J'ai listé mes cours ici. Voici quelques-uns de mes intérêts de recherche. Aussi, voici les incroyables étudiantes avec lesquelles j'ai le plaisir de travailler. J'aime étudier l'écologie et la physiologie des invertébrés marins et des algues. Aujourd'hui, nous allons examiner plus en détail le premier sujet de cette liste. J'ai découvert que je suis le plus heureux lorsque je travaille sous l'eau pour étudier la vie dans l'océan ou lorsque je fabrique de nouveaux instruments avec mon équipe pour mesurer quelque chose d'intéressant dans l'océan. À fin décembre, nous avons fait nos valises et déménagé ici avec un petit tackle qui s'appelle Max. Max pense qu'il est Napoléon réincarné et le chat diabolique, Monsieur Elliot, Monsieur Inscrutable. Ma femme Suzanne adore cette ville et c'est déjà lié d'amitié avec le consul général, Dr. Mélanie Zimmerman, car elle aime les chiens et sont experts en cuisine. Nous avons rempli une remorque avec mes robots et des autres choses à apporter au vieux Québec. Nous habitons sur rue Sainte-Anne, près de Frontenac. Veuillez visiter si vous êtes dans vieux ville. Après la vague d'Omicron diminue, je visiterai plus souvent le campus Laval. Comme je parle lentement le français, je pense que le séminaire se passera mieux si je parlais à l'anglais afin de pouvoir terminer le séminaire dans le temps que nous avons aujourd'hui. Mon professeur de français au lycée, Madame Shuffleton, elle disait que je parlais comme un écolier fou ou dingue. Donc, c'est probablement une bonne idée pour vos oreilles que je parle en anglais. Elle disait aussi que j'ai oublié mes mots de vocabulaire et les règles de grammaire. Puis, elle me jetait un morceau de craie à la tête. C'était un peu terrifiant pour moi. Les diapositives sont en français et maintenant je parle en anglais. So marine science has a défi persistant, a long-standing problem that's been with us since oceanography was born as a science on the HMS Challenger. I want to ask, am I covering up the bottom of the slide with the pictures of the attendance? Are you seeing that on your screen?
It's all okay, Mark. It's fine.
It's okay. Okay. So sampling takes money and time, and that constrains what we can understand about the ocean. Polar regions are a particular challenge, and most of the data that we collect, I'm trying to get the animation to play here. There we go. Most of the data we collect is because we sample at points. And that means we end up with a problem called aliasing or the crénelage. So qu'est-ce que c'est le crénelage? So very simply, because we don't have infinite time and money, we sample the ocean at certain points in space or time. That's these dots here. And the blue line is where we sampled, but the ocean may be doing something different. The red line is maybe what the ocean is up to, what nature is doing. So you can imagine, put your favorite variable here on the y-axis, and this can be space or time. And what do we do with all our data? We draw lines through them, just like your students when you teach laboratory. You connect the dots and you tell a story, and that story may be contaminated, or you may be fooled by Mother Nature. So what's a solution to aliasing crénelage? I am a big partisan for robots and sensors helping us. I like to think of autonomous robots as taxi cabs for sensors. They don't get tired. You don't have to pay them extra money if you make them work weekends. Of course, we don't do that for our grad students, right? Anyway, they can go places that we might find dangerous, deeper than we can scuba dive. Here are some of the robots that I have developed through the years. And I've also listed some behaviors there in the box below. So the challenge of sampling the ocean is even bigger in the Arctic. It's such a vast territoire. Here's an example from researchers, including those at Laval. This was a meta-analysis that Phil was part of that came out recently, looking at how marine plants, in particular things like kelp, eelgrass, how they may change as the Arctic warms. And while there is lots of hard collected data there, note the big gaps on the map. And as the ice goes away, as the Arctic becomes more ice-free with global warming, we will have more opportunities to fill in these gaps. And I am hoping that we can reduce the cost of the data we collect by using technology like robots. So why am I here? I'm here because you have Arctic Kelp hosted at Laval, funded by ArcticNet. Phil is the lead, and it was Lad who got me interested in all the fantastic work that is coming out of Arctic Kelp. I'm very intrigued with the ambitious goals, the objectives scientifiques listed here. This is a comprehensive program to understand what is happening with kelp in the Arctic and how things are changing, and to gather very important data so we can see these changes. I don't need to explain to this audience why kelp are so important, especially not to Professor Johnson and company, who are phycologists with a capital P. It's such an important species worldwide. It's a foundation species for an ecosystem, kelp beds. And it also is very important for food security for many people, not just in the Arctic. But in the Arctic, so many animals depend on kelp beds. And kelp was traditionally a food of great importance for First Nation peoples, and it's now coming back in popularity. There's a picture there of Maki Liebert, who's an entrepreneur in Greenland, who is starting to harvest kelp. And he's actually marketing to his own people in part, in addition to overseas. So there are many questions about how a changing Arctic will affect kelp as the temperature goes up and the light increases because there is a shorter ice-covered season. How will the kelp beds do? Will the melting ice, which is fresh water, lead to more turbidity? Could we have more stratification that could negatively impact the kelp? What will happen to the animals that depend on kelp, in particular, the main herbivore, the sea urchins? What will they do? So today, here's the plan de la journée. I'd like to start off talking about the technology and then change to a software technique, l'apprentissage automatique, that can help with the huge amount of data that this technology can gather. And then I will turn my attention to what I hope to do during my Bourse Fulbright with you, and some thoughts on the value of how our partners in the North, the First Nation communities, the Inuit, can maybe help us in this effort to understand these changes. So the technology. First, I'll talk about the types of robots, the smaller robots that I've developed that have a US patent, and then give some examples of how I've used them in the past. And then a new kind of sensor that my lab has developed for microplastics, which can also do double duty for measuring zooplankton. So the first robot I built back in the 1990s now was called Fetch, and it was a collaboration with my ami Jim Sias. Jim is an amazing person. If you've ever used a slide projector made by Kodak, he invented the Kodak custom carousel slide projector when he was 21 years old. It was Kodak's best-selling product ever. And I think he told me $100 as his bonus the year he designed it. Anyway, he's an incredible mechanical designer, and I partnered with him to make this robot Fetch. And I did all the electronics and computers, and he did the mechanics. So this project went very quickly. It was just a two-person invention. We privately funded it. That's code for, we used our bank accounts and our credit cards. And we achieved some firsts with it. We used a personal computer as the brains. It was an Apple Power PC. And at the time, the Power PC, you could not take it out of the United States because it was considered a supercomputer. It was so fast. So I like to say that Fetch was the world's first swimming and only swimming supercomputer, because shortly later the US government changed what constituted a supercomputer. We spent so much money that our families said, will we ever see that money back? Maybe you should try and sell these. So I founded a company with Jim that ran for 10 years in Virginia called Patterson. And we made robots, a handful of robots for government and academia. And then in 2006, we were acquired. And then I got technology back in my lab because the new owner left the AUV business. So William and Mary, my former employer, had the technology, and then it all came to Northeastern University when I moved there in 2014. So some examples of how to use this technology. First example involves water quality, and in particular oxygen. This is work I did with several people, most recently Anne-Marie Hulver, who's a doctorant élève at Université d'État Ohio. And we gave this talk, some of these data, at the International Coral Reef Symposium last summer. I'm very interested in oxygen because it really is the universal currency, not just of coral reefs, but of most marine ecosystems. And the scale of variation of oxygen is so interesting. It varies over many factors in proximity to organisms, as well as at the landscape or seascape level. And there's an interesting daily change in many ecosystems. The motivation for understanding coral or oxygen on coral reefs is that corals are so dependent on oxygen for growth and their health. We used as our framework a model by a Japanese researcher named Nakamura. And oxygen in the ambient environment outside the coral has to go inside, especially at night. And this is very important for understanding how fast calcification can take place deeper inside, away from the environment. So how did we study this? We used technology developed in my lab. We used handheld profilers to work in the boundary layer. And we also used the robot carrying the same sensors that we used in the boundary layer to examine what's happening. And we saw these beautiful dances of oxygen in the layer of water very close to the corals. And what we found surprised us. The dances were not nice, smooth curves. They had kinks in them, and they would move around in strange ways. And this was in the bottom boundary layer, so that prompted the question, what's happening at the landscape level? So we swam the AUV over the reef with the same sensor that was on the handheld unit, and we found these amazing large-scale patterns of changing oxygen that we would never have seen if we had sampled at the surface with a boat or only relied on a point measurement over the corals. So we found these large structures that have length scales that depend on whether you are going horizontally or vertically. And we characterize them from the AUV data set using spectral analysis. So what is happening here? What is causing these rather rapid changes? We think we have evidence that an instability in the boundary layer is occurring because there is enough stratification and the flow is strong enough over the reef that we get what we think are Kelvin-Helmholtz waves. You may have seen Kelvin-Helmholtz waves in the atmosphere. is shown in the picture upper right. You can see them in air as well as find them underwater. And this means that the environment around the corals can change very rapidly, and this will affect their metabolic rate at timescales that we formerly did not think were important. The paradigm before this was that reefs were well-mixed environments and that these sorts of things would not happen. Another example, we used the robot to look at rapid changes around a new kind of hydrothermal chimney in Eyjafjörður in Iceland. I spent part of my previous sabbatical there. Iceland's a fantastic place, like Quebec, for a sabbatical. It's a place where the mid-Atlantic ridge comes out of the water. That's my daughter, Serena, standing in North America, and across the road you can see is the European tectonic plate. My friend Jörundur Svavarsson was my host there at Háskóli Íslands. He's a marine ecologist who has done a lot of work with the benthos in shallow and deep water. And then my other friend, Daniel Jones, was my postdoc on this. He's now director of the Serpent Project, which is a fantastic project at the National Oceanography Center in the Gran Bretaña, and they use ROVs on oil platforms to do deep sea science. When the ROVs have downtime, the oil rig operators are fascinated with working with the scientists when they don't have to maintain the rig. Anyway, hydrothermal vents. There's 4 types of vents known, and only one of these special vents is found in Iceland. And you are probably familiar with the deep sea hydrothermal vents, which are very high temperature and acidic. And there are shallow water acidic vents in places like New Guinea and the Mediterranean and Taiwan. And in some of these places, it's been found that the water poisons organisms that can act as a food source for other organisms. And then in 2005, a new kind of vent was found in the ocean. That's a high pH vent, not a low pH vent. And this is a process where the vent is constructed through serpentinization. It's an abiotic process. And then in Iceland, around 2000, a new kind of vent was found in shallow water, one reachable by scuba diving, with high pH, pH around 10. So very basic water. And that's the vent we used the robot to study in Eyjafjörður, which is the fjord on the north shore of Iceland. It's the longest fjord there, and it was found by a submarine in 1998. pH 11 water comes out. The water is not saline, it's fresh water. There are weird microbes in the water, new species of archaea. And this is Iceland's first underwater park, which was part of the attraction. I think I have a little movie of, so it's a fantastic place to dive. The water is very cold, and then all of a sudden you see shimmering hot water. And if you put your hand in there, you'll burn yourself. The vents are huge. They're every bit as big as the vents in the deep sea. This is a multi-beam sonar image. On the west side of on the east side of the fjord, the vents are really tall and skinny. We worked on the other side of the fjord, which was a little easier for us to reach. And we asked these questions. What are the vents doing to the community there? Is there maybe bacterial subsidy like you see in the deep sea? Is there poisoning going on maybe with hydrogen sulfide like the vents in Taiwan? Are there more of these vents around Iceland? There's a diverse benthos there. It's very similar to maybe some of the benthic communities that Ladd and Phil have studied in terms of composition, many suspension feeders. The landscape is down the volume a little. It's a crazy landscape. These towers of this clay called smectite are everywhere. And you're looking at a landscape that's probably hundreds to thousands of years old, but it's a interesting oasis, not only for suspension feeders, but also small fish, juvenile cod and pollock come there. So we brought our same technology that we use for coral reef work there, the handheld instruments and the robot, and we made collections to see what the stable isotopes were around the vents. And we also used the handheld instruments and the robot. And the robot found interesting landscape changes similar to what we saw in Florida. So here's salinity swimming over a few hundred meters in the vicinity of the vent on the west side of the fjord. And we see huge changes over short distances where the salinity goes from 35 parts per thousand down to 18. And so that prompted the question, okay, are the zooplankton happy about that? So we did plankton toes, and we would stain our toes with a vital stain, neutral red, that my friend Cam Tang, who's now in Great Britain, developed. And we found that many of the plankton were dead over the vents. They were being killed by the salinity. So we put out sediment traps around the vents to see if there was a rain, a pluie coming down of dead zooplankton. And we found that the traps would fill up, sometimes even with larger decapods. So this was very interesting that this was a killing field for zooplankton over the vents. Linnea was helping my postdoc sort all of the invertebrates and algae that we recovered so we could do stable isotopes. And we found that most of the vent taxa are depleted in both nitrogen and carbon, but we don't understand exactly why. It turns out there is no bacterial subsidy that we can really detect. The horse mussels, Modiolus that we hope to use as a exposure meter for exposure to vent water did not work out as planned with their stable isotope signatures, but they did shift at distances away from the vent, but the ones right next to the vent, and this is very strange that this cold water mussel loves to nestle right next to the vent water. The ones closest had an isotope signature that was the other direction. So we are still puzzling over the data. This is one of the manuscripts I am working on here in View, Quebec for my sabbatical. So we hope to continue to work in this system. I want now to shift to the use of robots for mapping, and this may be of interest because we hope to do similar things with the kelp in the subarctic and Arctic. In 2008, I led the biggest expedition of my career. We had 18 scientists from 4 countries. We had 3 AUVs, and we went to the Caribbean, to Bonaire, which is Dutch Antilles. And this was for the International Year of the Reef. And there's an amazing data set that was collected in 1985 by a Dutch scientist named Fleur van Doel. And she mapped the entire side of the island away from the winds using scuba diving and aerial photography. And we wanted to see, okay, how much change has taken place in Bonaire? And can we more efficiently map the change with robots as well as with scuba divers? So my student Noelle Rellis, shown here, this was a good part of her PhD work. And she led the diving teams that did the surveys. And then we had several robots running around making maps. My late friend, who I miss very much, Zia Rahman, helped with the underwater video that we collected. So he developed an amazing machine algorithm based on a theory of color space called Retinex. And this allowed us to clean up our images. Watch for the sea turtle coming up here in a minute. These images, it's much easier to make identifications when you have better color and the picture looks clearer. There's the sea turtle. We also ran robots down deeper than we could visit through scuba diving. And these are some pictures of how we can make composite indices using things like the slope and the rugosity, and classify the bottom type using machine learning, and then make composite indices to look at diversity of the seafloor and make predictions about where the highest diversity will occur for the organism. So we made a spatial map, and we discovered that the mesophotic reef of value with high diversity is just below the limits of the marine park. So Bonaire is thinking about that information. We also were able to discover deeper on the reef that there were some mesophotic reefs that people were unaware of using the robots. Before we talk about machine learning, I want to talk about a new kind of sensor that came out of my laboratory. This is Ethan Edson with his invention. It's called Manta Ray previously. He now calls it the sensor, the capteur. He was an undergraduate at Northeastern, very talented, and he became intrigued with the problem of microplastics because he went on some voyages and he kept seeing plastic everywhere on his cruises. And this is a very severe problem for the entire world ocean because microplastics are ingested by so many organisms. And they aren't just at the surface where the seabirds and whales and some fish can get them. They become neutrally buoyant where the plankton can eat them, and even negatively buoyant where they end up in the benthos. I had a master's student, Molly Waters, and she studied what happened to the physiology of Middleus edulis when exposed to microplastics. And she found that just a short exposure can increase the respiration rate, and that elevation lasts for a month, which we had no idea was possible. So this is not good. All these effects combine together to impact marine ecosystems. And this is a problem we think, or could be a big problem for the Arctic, which tends to collect microplastics. Ethan has since co-founded a company, Ocean Diagnostics, here in Canada. And they are now making some technology that we have brought with us, maybe to use in the Arctic and subarctic during the field season. They make an imager called Saturna and a water profiler called Ascension, and we have both of these. My wife Susan knows how to use them. And they also have a data portal called Mariana, after the Mariana Trench. And you can see the data accumulating from users, but notice we need some data points in the Arctic. Just a bit about how Manta Ray works. It's or the sensor, as Ethan now calls it. It has a computer that sucks water in, and then there's a spectrometer that can look at the particles that are sensed and determine what kind of plastic they are, or even if they're not plastic, we can get an image. And it makes these beautiful pictures of plankton that you saw earlier in one of the slides at the same time that it measures the plastic. So we get lots of data with this technology. How are we going to analyze it? This is where machine learning can help. So I've got some examples here of how we've used machine learning. The first example uses side scan sonar, which is a method of seeing with sound. And we're one of the first, maybe the first robot to have side scan sonar on board. Normally, side scan is pulled from a boat, but when you use an AUV, you can get rid of the tether. So no more tether going to a boat, which makes the towfish go up and down and makes your images not so good. You can also get close to the bottom so that the sound makes very, very nice shadows like shown here. So these are some pictures. That's an airplane. Here's the bottom changing from sand ripples to a hard bottom off New Zealand. Here's what a close-up picture looks like. That's a tire from a car. And if you look closely on the screen, you can actually see the tread of the tire in the acoustic shadow. So lots of information in these images. Most of the time, people do not use side scan to look at things in the water column. They look at the bottom. It was developed to make pictures of the bottom, not pictures of the nekton. But I noticed that the images of the nekton, even though they were very blurry, had information. So we asked the question, can we use this information? So we took the robot to an aquarium. This was quite an adventure. The sharks, after a few minutes, it came over and started rubbing on the robot because of the electric fields. And we wanted to see, could we train a neural network to recognize the fish swimming by? And could we make an efficient network that maybe someday could fly inside the robot as it swam in the ocean? So a bit about how neural network works. They were inspired by real neural nets that organisms have. And if you remember your biology, the action potential occurs when there's enough excitation of the neuron by its neighbors. And we can make an algorithm that mimics this to a crude extent. So we have neurons that fire when they are given numbers. And this is called the input layer. And then we have a hidden layer, which has connections to its neighbors as well as the input neurons. And what we do is we adjust the connections so that only one neuron on the far side, the output layer, lights up when a certain combination, a certain vector of numbers is given on the inside. And so our task was to see, could we train and adjust these connections? This is what's fascinating about neural networks, is that they have a memory and they can learn and change as you encounter new things. We used a radial basis function as their architecture. A bit about the software that Fetch uses and why we wanted to do this. To my knowledge, Fetch is the only LabVIEW robot out there. LabVIEW is a programming language where you draw pictures rather than type lines of code. It makes it very easy to understand the software because you're looking at a picture as a very refined graphical user interface. You can also very easily make something called a state machine to control behavior. What's a state machine? A state machine is what is used on spacecraft that go to other planets. A state machine has states to deal with all the errors that may arise, as well as the work that must be done by the robot. And the definition of a state machine is that you can jump from one state to another state without having to pass through the intervening states. And it's very difficult to write a state machine when you type, but when you draw pictures, it's easy. So we made a state machine for Fetch, and we have a toolkit of software that I developed. And the nice thing about the software is once you've written something, you can make a picture that you wire to. So for example, here's a little algorithm that computes distance and heading between 2 points, which the robot needs to use. And we can make a toolkit, and we can combine these things into behaviors. So that's why I was able to make a robot with Jim Sias, is that the software allowed somebody who's not a computer robotics person to actually make a functioning robot. And the other thing about LabVIEW is that it's very good at handling the kinds of data that need to go to different payloads. And these are some of the payloads that our taxicat has carried. Anyway, back to the l'apprentissage automatique. So we were able to use LabVIEW to characterize the images it was seeing from the side scan, and with about 90% accuracy, which allows us to do useful work. And then we started experimenting with other taxa. So I was interested to see, could we actually see a jellyfish? The answer is yes, you can if you adjust the sonar properly. Can you see crabs swimming up in the water? Yes, you can. My kids were not enthused with this activity because I was putting crabs and jellyfish in our backyard pool to get imagery to train the network. We were invited by NOAA to go to the Antarctic to see whether this technology could help estimate and find krill, which are the base of the food chain there. Everything eats krill in the Antarctic. The biggest predators like leopard seals and the seabirds and the fur seals, everybody likes to snack on krill. This was probably the most exciting and difficult field trip of my life. The conditions were crazy. Launching and recovering the robot out of a small boat was crazy. The leopard seals were way too interested in what we were doing and would try and get into the boat, which could have led to death. The birds, the skuas, would attack the robot every time it surfaced, thinking it was a marine mammal in distress. But we gathered some important data. This little video here shows what it's like to swim through a krill swarm from the AUV's perspective. And this was important to get ground truth data of what we were seeing so that we knew that the side scan was actually showing us krill. And this is important to NOAA, who has one of our robots that they use for counting fish on the West Coast, because they can use the camera on their vehicle to know that they are seeing a certain species. And this allows them to calibrate a scientific echo sounder, which is what they use to estimate biomass. Okay, so last 10 minutes, what can we do together? Well, I obviously would like to fill in data gaps if I can, and maybe gather data on the menace of microplastics. And can we, augment the data sets that are already being collected? And can we use machine learning to learn things and reduce the labor of analyzing these data from the remote locations like the Arctic where you all work? And I want to give you some thoughts too on the value of citizen science, inspired in part by an experience I saw that Lad had with a École Inuit in Pond Inlet. We use Mathematica for a lot of our work in addition to LabVIEW. And recently we've been pivoting to deep learning, which is a different kind of machine learning that's very suited to image recognition. We use an algorithm called YOLO, you only look once, which takes a scene and divides it up into an incredible, crazy number of boxes looking for things. And then it does Bayesian probabilities on whether there's an object in the box. And then if there's something in the box, it computes a probability on whether that object belongs to a class of objects that you've already shown it. So I trained up a network that's available. There's a network in the Wolfram network repository. It's actually trained by a fellow named Julian Vincent, and I made some mods. And here's a picture of Max in Vieux-Québec, meeting an ami in the Dans la Rue. And of course, they're dogs, so they have to sniff each other. And the deep learning says, okay, there's a dog with this probability here in this box and another dog here. And oh, maybe there's 3 dogs here because the head is touching the rear end of the other dog. So that's one problem with these very capable networks, is they can be sometimes fooled. This is a problem that has been encountered by the Mesobot team that is trying to use an AUV to follow Nekton for hours and hours and hours. Sometime if 2 jellyfish touch, it thinks there's one, and then it follows the wrong jellyfish. But this is a very powerful technique, and it's now within the reach of all of us. You do not need a computer science degree to use this technology. The second bullet has a YouTube link that I encourage you to watch. If you'd like to do this yourself, you can actually make your own object recognition tool for very low cost or even free without having to type more than a line of code or 2. I have a little movie here that my friend gave me, Art Trembanus at University of Delaware, and he trained his iPhone to recognize crab pots. These are traps for crabs. And this is a big problem in the Chesapeake Bay because these pots get lost by the fishermen, and then they start killing organisms, crabs. And so he has money to use his AUV to find them and then go retrieve them off the seafloor. Anyway, this deep learning is now being embraced by the marine biology community. There's a great website listed here called fathomnet.org. And there are companies now that are capitalizing on this gold rush. I, in particular, like RoboFlow, which is a great company. They're the ones that made the video. So how can we put this all together? And can we enlist our neighbors to the north in this effort? So here's some pictures, absolutely amazing pictures from a blog from Arctic Kelp. And Lad and postdoc at the time, Karen Philby Dexter, were up there doing research, and they took some time and made a touch tank and introduced the kids at a elementary and high school, I believe, to the wonders of the benthos. And I absolutely love this picture here of the excitement about nature. We are all wired this way, especially as children, to appreciate nature. And I think this gives us an entree here into getting some interesting data. The benthos is not something that the Inuit normally encounter in great numbers. So there's an app for this that just launched 2 years ago called Siku by the Arctic Eider Society, and it's already got more than 9,000 Inuit users who have installed it on their smartphone. And Siku got money from Google Canada to make this, and the great thing about it is it's completely owned and controlled and partially developed by the First Nation communities, and they own all the data. This is very important term of use, is that they control the data. It was developed in part to get ice conditions, which are very important for the lives of northern communities, the people that live there, safety, hunting, food security, hence the name Siku, which is Inuktitut for sea ice. But because these people are so close to the land, obviously, they can enter and take pictures of the wildlife. And I was stunned to find that they now have a growing database of hundreds of pictures. Not many kelps, sorry lad. I think only 33 kelp pictures so far, but hundreds of pictures of different species of clams and starfish, obviously poisson. Anyway, this is a interesting opportunity. So Siku.org, which is the charity that runs this. Joel Heath is the head of it, and I've had some exchanges with him via email. Siku has a formal process for applying to use their data in partnership. Otherwise, you really can't get access to the images. So I am, of course, interested in kelp and the invertebrates, and trying to incentivize the northern communities to maybe take more pictures of the kelp that they see washed up on the beach, so we can start filling in the gaps, and then we can use machine learning to automate some of this. So I see I'm at the end of my time, and I just want to conclude with 2 more slides. I do other things, and if you are interested in hearing about Proteus, which is a nouveau laboratoire sous-marin that Fabien Cousteau is making, I have spent 89 days living underwater during my career on coral reefs, so I have lots of interesting insights. That's why Fabien asked me to help him design this new structure. I'm also interested in sciences des réseaux appliquées aux organismes coloniaux. And my longstanding interest is in ecophysiology avec les invertébrés et des algues. So, merci pour votre attention. J'ai hâte de vous rencontrer en personne. S'il vous plaît, abonnez-moi comme en québécois. Abonnez-moi sur Twitter et écrivez-moi mon courriel, texto aussi. Mon français est très rouillé. Pour que je puisse mieux comprendre, peut-être que les questions peuvent être posées dans le Zoom chat en français ou en anglais avec le micro. Et merci pour votre attention. Et I guess, Lad, are you there?
I certainly am, and I'm ready to manage the questions. En français ou en anglais?
Let me try and stop this.
Donc, vous pouvez réfléchir quelques secondes avant de poser des questions, mais j'aimerais dire bravo, Marc, pour ton français. Encore, c'est lent comme moi, avec un accent comme moi, mais c'est super. Et j'avais oublié de mentionner que Marc est doublement citoyenne canadienne-américaine grâce à sa mère. Donc, encore, Elle n'a pas appris son français ici au Canada, mais plutôt aux États-Unis avec Madame, le professeur, je ne me souviens plus du nom, Madame Cray.
I can't stop sharing the screen. Let me see if I can. Oh, here it is. Pardon, un instant, s'il vous plaît. Okay, wave control there. Can you stop screen sharing? Oh, here it is. Share. Oh, formidable.
Great. So maybe while waiting for the others to think of questions, I have one immediately, Mark, because I think that it's so important diving activity underwater, getting eyes under the water. And of course, you're working hard to replace those eyes or perhaps complement those eyes. No. So I'd like sort of your opinion about, well, and I don't want to get too political there, but as you know, the regulations for diving just get more and more severe. So sometimes, so where do you see this going, and what this dance that has to go between the old-fashioned Jacques Cousteau stuff and the modern Mark Patterson stuff?
Well, it's actually the modern Fabien Cousteau stuff, hence Proteus, which may be an interesting topic for discussion later. I truly believe human beings are the world's best sensors, captures. We have the best neural network on the planet, so we will always be the most important. When Proteus begins in 3 years, we hope, underwater in Curacao, we hope that robots and people will work together to explore this environment, including the mesophotic, where humans have less time. With Proteus, they will be able to spend more time, maybe 6 hours at 30 meters. And it's crazy what saturation diving allows you to do with respect to the time spent. So I think we will work together with our robot friends, is the short answer.
Okay, but maybe to just follow up on that, one thing that was very impressive was your spatial resolution of things, but the robots, there's a temporal aspect that's missing there. So how do you see, I was surprised not to see some sort of cables running all over the place as well, doing specific measurements for long, data loggers and things like that. Where do you see?
Yeah. Yes, we will have at Proteus, the intent is to have the entire reef wired up with sensors that are cabled. So we get temporal data as well as spatial data. That's extremely important to get a time series. I have a dream to have cameras and electrodes on the reef so we can investigate the life of plankton on a coral reef at a detail that has never been done. And microplastics, sadly, are a big threat to coral reefs because corals eat them like plankton. They get confused for some reason. They actually prefer to eat microplastics over zooplankton sometimes, which is very puzzling. Maybe there are chemicals coming out of the biofilm that forms that confuse the corals. So yes, of course, we will have networks of cabled sensors too, in addition to robots that swim like this here.
So I'm still waiting for questions, but in the meantime, so why did you choose, why was that site chosen? And again, how representative do you think that can be for what you are interested in?
You mean Curacao?
Yeah.
So Fabian and his team, and I've been advising him for many years now on site selection. We actually had sites in the Mediterranean and elsewhere. It wasn't just the Caribbean. And he has ambitions to have one in colder water in temperate zone, maybe the Gulf of Maine, maybe Canada. Canada has a long tradition of underwater exploration involving habitats, like subigloo. Dr. McGinnis, who's the leader in that space. We would love to see a habitat, for example, near a laboratory near a kelp bed. Curacao was chosen because there is a marine protected area there, and because Fabienne found a very rich person who gave a shore base land commitment to the operation.
Great. Okay, I see Jean-Eric has a question. Jean-Eric, allez-y.
Hi, Mark. Thanks for a great talk. That was really interesting. My question is, I think, is more related about the ease with which the community in general can use the wealth of data that originates from all these, sensors, AUVs that record a lot of data. And when we go on databases, usually this is the traditional format where, you have access to tabular data and so on. And when people use the data from these sensors, gliders, AUVs and so on, they will sort of use them in the way they want to and produce some graphics. But for the general community to access this data and be able to play with it in 3D for their own purposes, could you comment on developments for open source and how that data can be made available in some kind of a friendly way, I guess, to the general community?
This is a huge problem. The National Science Foundation in the United States wrestles with this. We now have to agree to deposit all our data in repositories, but that still does not visualize the data for you, et cetera. I think that really the solution are the young people. I tell all my grad students now that yes, I know you love animals and marine ecology, but oh, by the way, you are going to become a data scientist. you are going to be our expert at data visualization, and they rise to the challenge. There are incredible visualization tools in the R library. I'm a devotee of Mathematica in part because I can do nice graphs very easily. It would be nice, I agree with you, Jean-Eric, if the portal actually visualized the data for you to some extent, so you didn't have to go find your graduate student. Ocean Diagnostics with their Mariana cloud-based portal, they are trying to do some of that so that you may do quick visualizations of where the microplastics are and understand the size distribution and other characters. So I think this will become more and more common, but sadly, there is not just one magic tool, except your graduate student.
Thank you.
Speaking of graduate students, we have a question from one in the chat. Perhaps I'll just read it unless Matthew wants to come on board and say it. But great presentation. This is more of a technical question, but I'd be interested in hearing more about how sensors are integrated on a per mission basis in the AUV presented. For example, Fetch or the newest version, and the possibility for new sensor integration. Thanks. That's Matthew Huot.
Matthew, nice to meet you virtually. We've corresponded via email. Great question. So Fetch has a standard suite of sensors, and yes, when you add something new, you have to think about, okay, how will this work? And sometimes the easiest way is to package the new sensor, and this is during development, give it its own power source and data logger, and just strap it on the robot. So that's why I call AUVs our taxi cab. So we just use the robot's motor and its fins to drive around, and your payload does its thing, and then we come back and look. And that actually works for a lot of the things we do. It's only when we want to react to data in real time for anomalies, for example, that now we need to see the data as it's coming in. So many of the connections are up in the nose of the robot here. We attach things up there so they can penetrate and talk. I've also used the charging port back here. You can see a little black thing. That's how power comes in to charge the batteries, and that can do double duty. We can actually feed an instrument's data stream in through the port there. So there are several ways to do it. One last way that may be of interest to you, because I know you make instruments too, is because we have some places where there is not metal. Potentially you can use Bluetooth underwater to talk straight in through the hull to the computer, and maybe that could be the way. I hope that answers your question.
Great. Last call for questions. We're a bit past the hour, so I understand we're all in our virtual world needing to do other things, but sinon, amazing talk. Mark, I really enjoyed it. Merci beaucoup. Et comme on dit, Mark est là pour les prochains 7 mois. Donc, si ça vous tente de le rejoindre, en espérant en personne à un moment donné, mais par courriel ou par texto, même comme ça, vous êtes bienvenus de lui contacter parce qu'il cherche des idées. Phil et moi, on n'a pas rempli toute son dance card, donc on va sûrement avoir l'occasion de profiter de sa présence. Et
aussi pour les boissons au bar whisky.
Oui, la raison on est venu ici, c'est parce qu'on avait le café Full Élégie avec son collection de whisky là. C'est l'attraction du campus principal. Mais on espère, Marc, on se voit au campus prochainement. Mais sinon, on vous remercie une dernière fois. Merci pour la conférence.
Merci.
Merci beaucoup, Marc. Merci, Lad, pour l'animation et la présentation. Andréane, je ne sais pas s'il y avait des petits messages à passer avant qu'on se quitte, ou Debbie?
Non. Je pense que c'est beau. Et merci encore, Mark. Et we look forward to seeing you on campus and collaborating with you over the next 7-8 months. And on behalf of E&Q, thanks a lot. And for everyone else, watch our website and our infolettes, because we will be having another webinar next month. Okay, thank you.
Okay, merci
Bye-bye.
beaucoup. À tantôt.